So we've talked about mitosis. Mitosis is going to be primarily used for growth and repair. But now we're going to move on and we're going to talk about meiosis. Meiosis is going to be slightly different because our end goal is different. Take a moment, see if you remember, what is the ultimate goal of meiosis? What are we undergoing this process for? So meiosis is going to be the creation of haploid gametes. Gametes are going to be cells like eggs or sperm. They're going to differ from somatic cells in that they are haploid. They only have one member of each homologous pair of the chromosomes. And so their total chromosomal number is going to be 23. These are going to be formed by meiosis. Meiosis is a different process from mitosis, but you will find a lot of similarities. Meiosis is going to have two divisions that result in up to 4 haploid daughter cells. Ultimately, the function of meiosis is to keep the number of chromosomes in a body cell constant from generation to generation and help increase genetic variability in the population. So what we do with meiosis is we form haploid daughter cells. Those haploid daughter cells need to get together with another haploid daughter cell to create a new diploid individual. This is how we introduce genetic variation. We're also going to find genetic variation randomly sprinkled throughout the process of meiosis. It's pretty fascinating. This helps keep genetic variation in the population. I would love a discussion on why is that important? Why do we care about it? We had that discussion, or we talked about it very briefly way back in chapter 1 when we talked about reproductive methods. And some organisms reproduce sexually, meaning that they require 2 individuals in order to reproduce. This is going to introduce genetic variation. Some organisms reproduce asexually. And what are the pros and cons of each method? But we're going to move into meiosis. Meiosis is going to involve 2 cellular divisions, as we've already mentioned, and they have been so creatively named Meiosis I and Meiosis II. Meiosis I is set up to separate homologs. So when a cell originally has 2 chromosome number 1s and 2 chromosome number 2s and 2 sex chromosomes, Meiosis I is going to separate those homologs. Meiosis II is then going to separate the sister chromatids. Each division is going to have its own prophase, metaphase, anaphase, and telophase. So fortunately, we do not need to learn a whole new set of vocabulary. We just are going to have to apply it slightly differently. So if you can remember prophase, metaphase, anaphase, telophase for mitosis, you should be able to do the same for meiosis I and meiosis II. So we're starting off with those gametes. Oops, this slide is a duplicate. There we go. Meiosis has the 2 cell divisions. Again, more duplicates. All right, starting off with meiosis I. Oh, I'm going to back up just one moment to this slide. This slide is going to give us a nice overview of meiosis, keeping in mind that this does not show everything. It just gives us a general overview of what's happening. In meiosis I, we start off with a diploid parent cell. So you'll see that this diploid parent cell has 2 copies of chromosome 1 and 2 copies of chromosome 2. Once we undergo meiosis I, we're going to separate those. So here are 2 chromosome 2s are lined up, 2 chromosome 1s are lined up. We're going to split. Now each cell has 1 chromosome 1 and 1 chromosome 2. 1 chromosome 1 and 1 chromosome 2. So we have gone from a diploid cell to a haploid cell, which is great. That's half the battle, but we still need to separate these sister chromatids. And so we're going to separate out those sister chromatids in meiosis II. We're going to form daughter chromosomes, and now we have 4 unique cells, 4 unique haploid daughter cells. All right, so we're going to look at depth at meiosis I. Meiosis is going to be preceded by interphase, during which each chromosome is going to be copied and now consists of 2 attached chromatids, just like interphase before in mitosis. This process of meiosis I is sometimes called reduction division because it's producing 2 haploid cells. We started off with a single diploid, now we're ending up with 2 haploid cells. We are reducing the number of chromosomes as we divide. Each daughter cell has 23 chromosomes, 1 member of each homologous pair, and each chromosome is going to consist of 2 sister chromatids, which will need to be divided out before we are done with meiosis II. So starting off with prophase I. In prophase I, our chromosomes are going to condense down. Homologous chromosomes are going to pair up by a phenomenon known as a synapsis. Now here is something interesting that happens in the synapsis of our of meiosis. In the process of forming a synapsis, we're going to see what is known as a crossing over event. A crossing over event is a phenomenon by which we can introduce even more genetic variation into the population. And since we are going to cover it a little bit later in this chapter, I'm not going to go into it right now, but I do want to just mention that at prophase 1 of Meiosis I, we're going to see crossing over events at a synapsis. And like I said, we'll touch on that a little bit more later on. Metaphase 1, we're going to see matched homologous pairs line up at the midline of the cell and attach to the spindle fibers. This is where we're going to see another introduction of genetic variation known as independent assortment. And again, we'll come back to that. In anaphase 1, it's going to proceed just as you would expect it to. Homologous pairs of chromosomes are going to separate and move to opposite ends of the cell. Each homologue is going to consist of 2 sister chromatids at this stage. Telophase 1, the nuclear envelope is going to reform around those chromosomes and at the end of each cell. Cytokinesis is going to occur and we're going to end up with 2 haploid cells. After that, we don't go into a phase or a stage of interphase, because keep in mind, our process of meiosis is not yet done. We still have a whole nother round to go. So we're going to start off, between Meiosis I and Meiosis II, we're going to undergo a process known as InterKinesis. During InterKinesis, this is going to be a brief interphase-like period before Meiosis II. However, it's not quite interphase because there's no replication of DNA. Right now, all of our haploid cells have the correct number of chromosomes, but they still have duplicate copies of those chromosomes, which is not okay. And so what we need to do is we need to separate those sister chromatids into daughter chromosomes. So we're going to undergo a process known as Meiosis II. In Meiosis II, we're going to have prophase 2. During prophase 2, the chromosomes are going to condense. In metaphase 2, the chromosomes are going to line up along the midline of the cell. In anaphase 2, the centromere holding 2 sister chromatids are going to separate. The chromatids of each new pair are considered chromosomes, and those chromosomes are going to move to opposite sides or opposite poles of the cell. Then we go into telophase 2. In telophase 2, the nuclear membrane is going to form around the chromosomes at each end of the cell, and cytokinesis is going to occur in both daughter cells, forming 4 haploid daughter cells. The important feature of Meiosis II is that it separates 2 sister chromatids from each chromosome. So Meiosis I is separating homologous pairs, and Meiosis II is separating sister chromatids.
A cell that undergoes mitosis produces daughter cells that are dramatically different from the daughter cells produced by a cell that undergoes meiosis. Let's compare these 2 processes, beginning with a look at mitosis and Meiosis I. In both mitosis and Meiosis I, cells begin with replicated chromosomes, each cell having 2 chromatids per chromosome. In Meiosis I, the homologous chromosomes pair with each other and come to the middle of the cell together. In mitosis, the homologs move independently of each other. In mitosis, the chromatids separate. In Meiosis I, the homologs separate. In mitosis, a diploid cell divides to produce genetically identical diploid cells. Each cell contains 2 complete sets of chromosomes. In Meiosis I, a diploid cell divides to produce genetically unique haploid cells. Each daughter cell now contains just 1 set of chromosomes. Meiosis I is known as a reduction division because the outcome is a reduction of chromosome number. These 2 cells are daughter cells produced from Meiosis I. Let's use just one of them to compare Meiosis II to mitosis. A cell entering mitosis is diploid, with 2 complete sets of chromosomes. A cell entering Meiosis II is haploid, with just 1 complete set of chromosomes. In both cells, each chromosome has 2 chromatids. In both mitosis and Meiosis II, the chromosomes migrate to the middle of the cell. Then, the sister chromatids of each chromosome separate to form independent chromosomes. In mitosis, a diploid cell divides to produce genetically identical diploid cells, each with 2 complete sets of chromosomes. In Meiosis II, a haploid cell divides to produce genetically unique haploid cells, each with 1 complete set of chromosomes. If we consider that Meiosis II actually began with 2 cells, then the result is 4 genetically unique haploid cells. In both mitosis and Meiosis II, the division is not a reduction division. Only in Meiosis I are the number of chromosome sets divided by half.
So that's our overview of meiosis. I do want to share this diagram with you. This diagram is going to separate out the processes of Meiosis I and Meiosis II. So spend a little bit of time with this diagram. You should be able to, cover up the words, the text up above and below, and be able to identify each one of these phases on site. If on a final or a homework assignment, I may give you one of these images from a particular phase and ask you to tell me, is this mitosis or meiosis? Is this a mitosis or meiosis? If it's meiosis, is it meiosis 1 or 2? What phase of meiosis is it? Is it prophase, metaphase, anaphase, telophase, or in cytokinesis? So you should be able to identify each one of those phases based on what you're looking at. I will tell you, if I give you this question, I will tell you what the original number of chromosomes is. So I may tell you this particular organism has 4 chromosomes. Its diploid number is 4. And what you're looking at here is a haploid cell or a diploid cell or wherever. So I will tell you how many chromosomes that organism is supposed to have in either its diploid or haploid form, and you should be able to infer the rest. Please let me know if you have any questions, comments, or concerns. So we're going to continue on with our discussion of meiosis. Following meiosis, changes in shape and function of the haploid cells can result in functional gametes. We're going to see that there is a difference in the functionality of the gametes based on whether we're looking at female or male reproduction. Spermatogenesis is going to produce 4 sperm cells that are specialized for transporting the male's genetic information to the egg. Oogenesis produces up to 3 polar bodies and only 1 ovum packed with nutrients to nourish the early embryo. If you remember, go back to our previous chapters when we talked about reproduction, and we talked about the difference between male and female. What's the term? Reproduction methods or goals, I guess. There's a better term for it, and my mind is blanking. But the male sexual strategies, there we go. Sexual strategies for males is going to be to produce as many and as fast sperm as they possibly can. The sexual strategy for females is to produce 1 super healthy, prime, ready to go ovum that as soon as it receives the additional copies of genetic information, it's ready to roll. So here we have sperm formation, and we have egg formation. And they're going to undergo slightly different processes depending on what their ultimate strategy is. But the strategy for the spermatids is going to be to produce super light, super fast spermatozoa. So these sperm are going to have little more than chromosomes and mitochondria. Really, that's all they have. They're not going to spend a whole lot of time on anything else. Whereas the egg is going to be the one responsible for containing all of the cytoplasm, all of the nutrients, all of the mitochondria, to nourish the zygote. Yeah, I believe that's it for this section. The last thing that we're going to talk about, no, I'm going to pause here and we're going to come back and finish up this section in just a moment.